All right, back to glycolysis. Now that we sort of have the introduction going, we're actually going to really look at what happens to the glucose. So remember, glucose is in the cytoplasm. And now we'll look at these steps. I'm not going to expect you to remember all the details in the next couple of slides, but I think you have to look at the molecules and see the structures to appreciate what's going on. And there are a few key ones you have to remember. As an overview, what's going to happen in the cytoplasm is glucose is going to come into the cell. We're going to use up 2 ATP, which seems kind of counterintuitive. Why would you use energy to make energy? But it's sort of like that old saying, you spend money to make money sort of thing. We're going to invest a little bit of ATP, but it'll end up coming out ahead because we will make 4 ATP in the energy payoff phase. And we'll end up with these things called 2-pyruvate and a little bit of water, a little bit of hydrogen ions, and then our friend NADH, that energy carrier, is going to come in and deliver that energy into the mitochondria as well. So let's jump right into some of the structures. Make sure, nope, I do want to go back here, sorry. So we'll look at it in a linear fashion, and I want you to remember every place that there's a number, an enzyme is making this transition happen. Although these steps are energetically favorable, they give off energy, they are technically spontaneous, they do not happen without the help of an enzyme. Do you have to know the names of them? Nah. It's worth knowing a little bit of recognition, having a little bit of recognition. We know enzymes always have an ASE ending on them. A hexokinase takes something and adds it to a glucose, which has 6 carbons, which is where your hex comes from. And then kinases are going to come up many times. You've already heard about them in cell signaling. Kinases tend to move phosphates to a substrate. So the first step, glucose is going to literally get on its way to getting trapped inside the cell. You're going to invest that ATP. Remember, phosphates have a significant amount of charge in them. So this makes this glucose molecule here, when it has that phosphate on it, now it's called glucose 6-phosphate, because the phosphate's added to that 6th carbon. 1, 2, 3, 4, 5, 6. It gives it some negative charge, which keeps it in the cell better. It will not go through its transporter to leave the cell, so you kind of force it down this pathway. Phosphoglucoisomerase is going to move a bond. Remember isomers? So glucose and fructose are isomers. It's moving a bond, and now we will now have fructose 6-phosphate. Then we're going to invest another ATP, and this enzyme is worth thinking about a little bit, phosphofructokinase. So it transfers a phosphate to that fructose, and this is an enzyme that is regulated. So this is the one that will have a feedback loop. And if there is lots of ATP in the system and we don't need to go down this route, phosphofructokinase shuts down. One reason to think about that name. Okay, so our whole goal in this first half is not only to trap the glucose in, but also make it symmetric. So look at this molecule, this fructose 1,6-biphosphate. Again, you don't have to know the name so much, just recognize it. It's very symmetric. Except for that OH hanging up and this guy hanging down, it's almost the same thing. So what's going to happen is we're literally going to cut this in half. And that's what happens in the next step. We have 2 3-carbon things. These guys, again, are isomers. They're exact same atoms, but just hooked up in slightly different arrangements. And we are going to end up using this one, glyceraldehyde 3-phosphate, or G3P. So the names that you need to know in this system, glucose, you don't have to know so much these, if you recognize them. But glyceraldehyde 3-phosphate, I do want you to know. This molecule is going to come up when we talk about photosynthesis. This is the first sugar that's made that ends up going into making glucose and fructose for your in the plants. Okay, so now we need to talk about something called substrate level phosphorylation. And this is how we are going to make some energy in the payoff phase. So we are not going to put our molecules through any kind of a fancy pump, which I'm going to talk about in a few minutes, but we are just going to take an enzyme, or an enzyme is going to take your substrate, rip that phosphate off, add it to ADP, and voila, we will have an ATP. And that's the big picture that you need to know happens in the second step, the energy payoff phase. Steps 6 through 10, we take our glyceraldehyde 3-phosphate, which adds 1 phosphate on it. Also remember, this step is going to happen twice, right? Because we made 2 of these up here. So eventually that molecule converts into G3P, and we'll do this thing twice when we do our accounting later. So we're going to take this molecule, Move bonds around. Every time you move bonds around, you can take an NAD and reduce it to NADH. So again, that happens twice. Then we're going to take this molecule, and because these bonds phosphates are never very stable, an enzyme can come in here, lower the energy, and rip off those the phosphates and make some ATP. This is a substrate level phosphorylation event. An enzyme rips that phosphate off and puts it onto ADP to make an ATP. Again, more movements of bonds, moving that phosphate around, again, trying to make it unstable near that oxygen. It's not very happy energetically. And now an enzyme can come in and rip off the phosphate. And we are left with producing a total of 4 ATP, 2 NADHs, and then this molecule called pyruvate, which you probably do need to know the name of because we're going to keep talking about it. So here we are back in our big picture. Glycolysis just took place. We made some NADH. We made a couple of ATP with substrate level phosphorylation, and now we have 2 pyruvates, which are going to go get imported into the mitochondria. Then that 2 carbons that come in are going to go through the citric acid cycle, and then we're going to, again, rip off some energy to have it go through the electron transport chain. It sounds like we need to get my puppy dog to stop barking. Okay, so now we are in the mitochondria, and we are going to talk about the citric acid cycle. It's also known historically as the Krebs cycle because Sir Krebs is the man who figured out all the enzymatic steps, which is a huge feat biochemically. I tend to slip into that nomenclature just because that's how I learned it, but it is called also the citric acid cycle, and you'll see why. So let's look at it kind of schematically, and then we'll dig apart a few more details again. So remember, pyruvate is formed when we finish glycolysis. It has 3 carbons. We have these nice little gray balls to represent our 3 carbons. That gets imported into the mitochondria through a special fancy transmembrane complex. It's 60 different proteins make up this complex. It has to span 2 membranes. That's one bilayer, and that's another bilayer. As it comes through, and this picture is not really appropriate because it happens actually right on the membrane, those 3 carbons, 1 carbon gets ripped off. One thing I'll say over and over again is every time a bond gets broken, we want to capture that potential energy, and it's going to get put into this reduction of NAD to NADH. Coenzyme A is literally that, a coenzyme. It's not protein, it's a molecule, it's an organic molecule, and it's going to help shepherd those 2 carbons into the citric acid cycle. It's called a coenzyme because it gets reused. It never gets used up, very much like an enzyme. And then those 2 carbons are going to float around. They're going to get attached to a 4-carbon thing to make a 6-carbon thing. We'll see in a minute. A couple more carbons will get ripped off in another place. Again, anytime you break bonds, you make an NADH, blah, 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 make a little ATP. And then it's a cycle because we're going to regenerate the starting material. Oh, should have known I was going to do that. So let's dive into the structures just a smidge. So 2 carbons come in. Again, I want you to look at this for the level that it fascinates you, and you don't have to remember the carbon skeletons. You just have to know in general that you're going to take 2 carbons, attach it to a 4-carbon thing. I think knowing that name is worth it. It's called oxaloacetate. So acetyl-CoA drops off its carbons, an enzyme. Every time there's a number here, there's also an enzyme there, but we won't look at their names this time. And we now make a 6-carbon thing called citrate. This can gain and lose a hydrogen. If it was just in isolation, it would be citric acid, and that's where the name comes from. Citric acid is the stuff that makes your orange juice tangy, your lemon juice tangy. The acid part of it does that. Again, I'm going to kind of fly through what's really going on. The big deal is that several times we are going to rip off a CO2. Again, for those of you guys who love the biochemistry like me, you can color code and watch these carbons. So the 2 carbons that come into the cycle are not the carbons that leave the first time around. You actually start losing these carbons as CO2. So we say we exhale CO2. This is where you get it from. Those carbons get ripped off in the citric acid cycle with an enzyme. Again, every time, and this one's going to go here, every time you break a bond, you transfer the energy to NAD to make NADH, which is going to go the electron transport chain later and make some energy. So we're going to rearrange. Now we have a 5-carbon. rip a carbon off again, transfer the energy, make some CO2. Now we have a 4-carbon guy. This is one place where we make an ATP. This is again substrate level phosphorylation. The rest of the time, then we make this other guy, FAD. He's sort of a relative of NAD. It carries a slightly less amount of energy, and we're going to see how it interacts with the electron transport chain that way later. Again, rearrange some bonds, rip apart a bond, move something around, capture it in NADH. The bottom line is that every 2 carbons that come in make 1, 2, 3 NADHs, 1 FADH, and 1 ATP. First it makes a GTP, then it transfers it to ATP, but again, you don't have to know that name. It's a cycle because you regenerate that starting oxaloacetate material after you go through. And now those 2 carbons that here, they basically have become part of that chain, and now they're going to get recycled. So just to remind you again, substrate level phosphorylation is a slightly inefficient way to make the ATP. It's what happens at the bottom of the citric acid cycle. You rip a phosphate off and add it to ADP to make an ATP. So then what happens to the rest of the energy? That NADH, FADH2 is going to come into this big thing called oxidative phosphorylation, which is the electron transport chain in chemiosmosis. And this is where we're going to make the most ATP. This all happens on the membrane of the mitochondria. And this is a great slide. It's in your textbook to kind of do some accounting. We will talk about this in class, so I'm not going to spend too much time on it now. And in fact, we will pause here.